Hello, my name is Jason Linville, and this is a lecture on the polymerase chain reaction recorded for CJ352 Forensic Science Lab 2. So this is an overview. So we're going to have a couple, the polymerase chain lectures. There's, I believe, 4 of them. And this first one is just sort of an overview of the process as far as what PCR is, what the polymerase chain reaction is. In some lectures on the syllabus, the lab may be DNA amplification. It's the same thing. The polymerase chain reaction is the process used to amplify DNA. So sometimes those terms, DNA amplification and PCR, are used interchangeably. So what we want to accomplish is there's an approximate number that I'm going to give you that might not always sort of apply depending on the exact extraction, but I want you to sort of just have this approximate number in just the concept of how many copies of DNA we're starting off with versus how many we're finishing with after we amplify DNA. So we can sort of see the start and finish lines there. I also want you to be able to name the 4 PCR components, and then we're going to go into some more details of those components in a later lecture. I'll list the steps in a PCR reaction. So a PCR reaction is sort of like a step-by-step process. And notice reaction here of what's happening on the molecular level. So not what the human is doing or the instrument's doing, but what's happening on the molecular level. So it's a little bit difficult to understand. And then finally describing the order that the PCR components, the 4 that we're going to go over, the order that they participate in the reaction. Yeah, that's a little bit better. Okay. All right. So again, to summarize, where we're starting off with is throughout this entire course, the concept is we find blood at the crime scene and we're converting it into this DNA report. Okay, so we've gone over the collection process. Okay, we're going to collect this on a swab. It goes back to the lab. We take a little bit of this and do a TMB presumptive test, right? We determine it's blood. So then we snip off the entire swab or cut out a portion of the stain, put it through the extraction process. And this is where we're at in the course. We've gone through this silica extraction process where we end up with a tube about the size of our thumb. And it just looks like it has a drop of water in it. It's a drop of TE buffer. It is essentially a drop of water, but it has the DNA dissolved in it. Okay. So we want to sort of know, okay, how many copies of DNA are we starting off with? And the amplification process, we're going to take a little bit of this DNA, put it in a smaller tube, okay, and then put it in an instrument to amplify the areas of DNA we want to amplify. Okay, so that's sort of where we're at in the process. And we're still not, after this amplified DNA, then we put it into another tube and eventually it goes into the big instrument that spits out this report. Well, spits out data that then is converted into the report. So we're still a ways from the finish line here, but we're just going through over the amplification process is what's next. Okay, again, we're trying to get here. These 13 pairs of numbers. Okay, that's what we're describing this at. Well, we're going to amplify in this reaction 13 areas of DNA. But before we get into the details of how to amplify these specific 13 areas, we sort of want to back up and just talk about the general process of amplifying DNA. Okay, now notice here, this all DNA, this is our extraction. This is where we're at right now. Okay, and where we're going right now is into this area, 16 segments of amplified DNA. But notice here, thousands of copies, 10 million copies. Okay, in our tube that you just finished extracting, there was probably about 1,000 cells in the sample. And again, this really depends. There's probably a lot more cells in the buccal swab than in the blood stain. In some evidence samples, there might not be nearly as many as 1,000 cells, but we just want to sort of thousands of copies, okay, of DNA. We're probably not in the millions of copies. We got to be a little bit above, the hundreds of copies in order to get this to work. So thousands of copies is sort of the ballpark of where we're at. So we want to go over how do we take those thousands of copies of DNA, thousands of cells, essentially, we busted them open, and now we have thousands of copies of our DNA. Okay. Remember, 1 copy of each allele in each cell. Okay. 2 copies of the genome because you get 1 copy from your mother and 1 copy of your father. But we don't care about the details here. We just want to sort of the general numbers. Okay. So after extraction, we have about thousands, and now we're on this step here, amplification or polymerase chain reaction, which is the method for amplification. We'll get to this stuff a little bit later. Okay. All right. So after DNA extraction, we have thousands of copies in that drop. I know, Dr. Linwood, you said that about 5,000 times. Hey, it should be easy on the question on the exam then, right? All right. Now, here's the extra piece of information I want you to get from this slide. Thousands of copies of all of the DNA. We open up a cell and all of the DNA, all of the pages of the recipe book, it just all spills out into our solution. Okay. But remember, 99.8% of that is exactly the same among individuals. We, as forensic scientists, don't care about that. We're not trying to amplify all of the DNA. We just want to pick the pages, the paragraphs of the recipe book that we know are different among individuals. And we just want to amplify those areas of DNA, small areas of DNA that are maybe like, 300 base pairs long or something like that. That's all we want to amplify. Okay. So at the end of this amplification process, we're going to amplify the one small area. Okay. Except we're going to amplify like 13, 16 of them at a time. In class, we're going to pick 3 of them at a time. That's not important, but it's just a small area and we're going to make a copy. So if you imagine this as a little magic school bus situation, we go into that DNA extraction, all of this thousands of copies of DNA of all of it, all 3 billion base pairs is floating all over the place. We find the area that we want to amplify and we make 10 million copies of that area of DNA. Okay. So we go from thousands to 10 million copies. We go thousands of copies of all of the DNA to 10 million copies of one small area. All right. So hundreds, and again, sometimes I use hundreds, sometimes I use thousands because we probably put hundreds in the two, but there's thousands there total. So, but if we just tried to somehow label that DNA that was present in our extraction and measure it with an instrument just by chopping up the DNA, the problem is that's a very low signal on our instrument. We can't really see the DNA that's there or measure how long it is. We need millions of copies before we can actually get a peak or some type of reading that we can actually see on our instrument. That's why amplifying the DNA is such an important step. Okay. And there's other reasons as well, as far as you sort of amplify the area you want, and then the other stuff that's there doesn't really matter too much. But we can't just chop up our DNA. I mean, that used to be how they did DNA analysis. They would extract it, chop it up, smear it out, and label it with a probe and sort of try to measure the DNA that way. It's very difficult to do if you have an evidence sample that does not have that much DNA in it. Okay. So this amplification just makes it a lot easier to measure the DNA. Okay. So when we amplify DNA, this is what the final product is going to look at like. But again, we're doing the amplification. It still needs to go into another tube and then into a big instrument, and the instrument spits out something like this. So we're still a couple steps away from this. But that last slide of that little bump there versus a tall bump, this is what we're talking about. Okay, we need the peaks to be high enough for us to read and see what's there. If you look closely at that other line, and I don't know how well this is going to come through on the recording, but it's like a whole bunch of little bumps is the baseline. So if these peaks get too small, we can't see them. Okay. We need them to be bigger than the noise in the background. So we can say, yeah, there's definitely a DNA there. Okay. And that's definitely a 9 allele and 11 allele. That's a 911. There's one of our pairs of numbers there. Okay. So how do we amplify DNA? What is this process? All right. PCR, the polymerase chain reaction. Okay, synthetic replication of a small area repeated over and over. PCR, in order to amplify the DNA, we're going to find that because of the primers we use, you need to know a little bit about the area of DNA and the sequence of the DNA. This used to cause some limitations, maybe in some animals, or if you're trying to amplify like dinosaur DNA, this may be an issue. But now that the human genome is sequenced and we know so much about it, we can really, scientists can get a good idea of how to amplify any area of the human genome that we want to. Okay. But PCR, it's not as though you can just throw DNA in a tube, run PCR. You need to know a little bit about the DNA you're trying to amplify. Lucky for us, we're trying to amplify human DNA, and we know a lot about the human genome. Okay. Learning objective number 2, or whatever we're on, where I want you to list the 4 materials in the PCR reaction. Okay, this is it, DNA. This is the DNA in our extract. Okay, so you already know where that comes from. You know a little bit about what DNA is. Now we got three weird words: primers, DNTPs, DNA polymerase. This lecture and also the PCR components lecture, we're going to learn more about what these three things are, okay, and how they play a role in the PCR reaction. DNTP, you can also think of those as the letters, the A, G, C, and T's in there. Okay. All right. So let's say that this is our DNA. Okay. So it's a ladder, you know, A is opposite T, G is opposite C. Okay, T is opposite A. So we've taken the ladder and we've just turned it on its side. So if we were listing the DNA sequence, we would only have to list, you know, one side of those. But we're looking at the DNA molecule. We want to understand what's going on at the molecular level here. Okay, so we have our DNA. And in order to copy this DNA, we need some type of machine. We are going to use a small biomolecule that's a machine that does something in the body. Okay, but for now, let's just imagine this like magic school bus type thing. We have a machine in here. And I think for my example, I used former Vice President Dick Cheney. Not really a machine. So I'll probably have to replace this soon because, as you know, from 110, anybody I use in a lecture tends to pass away eventually. Anyhow, so let's imagine we could shrink down Dick Cheney. He could get in here and he could see the letters A, T, G, C, T, A, C, G. So he just gets G's and C's and T's and A's and he just makes a copy of it. Okay. Okay. Well, there he goes. Makes a copy of DNA. Okay. This is one way to do it, sort of to read the DNA and assemble a whole new copy of DNA right next to it. Okay. But that's not exactly how it works. There are sort of several steps in this process. So let's go back. Let's keep our machine, but let's do it a little bit more accurate. Okay. The first thing we're going to do at the molecular level is separate the strands. Okay. Does everybody see what happened there? Okay. We separate we break the ladder into two in the middle of the rungs and we separate the strands. Okay. Now we have two machines that are going to come in and read each side of the DNA. Okay. And this is actually what happens on the molecular level. We have some type of polymerase. Okay. Reading, oh, C, I'm going to add a G here. Oh, A, I'm going to add a T here. Okay. And you actually need single-stranded DNA to be the template. And you can imagine now it's a lot easier where this molecule can just sort of run along. We got two of them, one on one side of the ladder, the other separate identical machine on the other side of the ladder. They're just reading the DNA and making the copies of it. Okay, this is closer to how it happens. DNA separates, and then a machine, okay, an enzyme, a protein, if you will, is going to be, remember, those are the little nugget machines that do something in the body, are going to read one side of the DNA and make a copy of it. Okay, but we're leaving out some steps here. So let's go back here. All right. The machine, before it can attach to the DNA, because remember, we're just looking at like 10 base pairs here. When we separate the strands in an actual PCR reaction, we have those thousands of copies separated, and they are just single strands, long pieces of DNA all over the place. And the machine that's going to make a copy needs to find the area of the DNA, those hundreds of copies, base pairs that we need to copy for forensic science purposes. We're not copying all of the DNA, just a small area. So the way that the polymerase locates the areas, they have a little bit of help. Something called a primer, okay? And that's the AGT there and the AGC here, okay, circled in the red box. Those primers are put into a reaction. Our second component, okay, we have our DNA in here. Then we have our primers in here. We put in primers that will attach to the DNA. And based on the sequence of the primers that we can control when we're squirting them into the tube, we order primers of a certain sequence, squirt them into our amplification tube. They're only going to attach in specific areas of the DNA. So we get one to attach at the beginning of where we want amplification to, and the other to attach at the end of where we want amplification to stop. Okay. And we sort of start at one end, start at the other end, and make a copy of either side of the DNA. Okay. So those primers are the second thing that we need in a PCR reaction. Okay, DNA primers. Okay. Now, it's not Dick Cheney, obviously, is a machine. So the machine, the biomolecule, the protein that we use to amplify DNA is called DNA polymerase. Okay. And eventually we're going to find a more specific name for it is Taq polymerase. So I want you to recognize that these two things are essentially the same. Taq polymerase is a specific type of DNA polymerase. Okay. But the polymerase is what is going to be able to, oh, okay, I need an A here. Okay. Then I need to put a T here. Okay. So we got the DNA component is 1. We got the primers over here. Okay. That's 2. Okay. We got the polymerase. Okay. DNA polymerase here. That's our 3rd component. And our 4th component that we need for PCR reaction, DNTPs. Okay. Those are the letters. So those are the G's, C's, T's, and A's that are all floating around. And when the polymerase needs a T to match up this A, it's floating around in the solution. It just grabs it, not really grabs it, but the T comes in and attaches to the A. Okay. So that's what I want you to get out of this lecture, sort of those 4 components. Okay. Now, what causes the DNA to be separated? What causes the primers to attach? What causes this DNA polymerase to be attracted? Okay. And then how do we get it to go through those cycles again? We do that through temperature changes, which we're going to talk in the next lecture. But for this one, I just want you to understand those 4 components and know the order. DNA separates, primers attach, that's 2. Polymerase attaches, that's 3, adds the letter, that's the 4th component. Got it? Review it. Okay, let's move on to the next one.